Bulk transport. Cells routinely import or export large molecules across the plasma membrane. Macromolecules are ingested into the cell from outside through phagocytosis and endocytosis. The opposite of endocytosis is exocytosis, where macromolecules are secreted out from the cell. Exocytosis is the outward transport of substances by carrier molecules and is quite common in secretory and excretory cells. This process is called cell vomiting or amniocytosis. In all eukaryotic cells, secretory vesicles are continually carrying new plasma membrane and cellular secretions like proteins, lipids, and carbohydrates from Golgi apparatus to the plasma membrane or to cell exterior by exocytosis. Proteins to be secreted are synthesized on the rough endoplasmic reticulum, RER, and passed into the lumen of the endoplasmic reticulum, glycosidated and transported to the Golgi apparatus by endoplasmic reticulum derived transport vesicles. In the Golgi apparatus, the proteins are modified, concentrated, further glycosidated, stored, and finally packaged into vesicles that pinch off from trans-Golgi tubules and migrate to plasma membrane to fuse with it and release the secretion to the cell's exterior. During exocytosis, the vesicle membrane is incorporated into the plasma membrane. The amount of secretory vesicle membrane that is temporarily added to the plasma membrane can be enormous. In this process, cells having secretory function, such as pancreatic cells, pass out their enzymatic secretions outside the cell. Sometimes, the large-size solid food of foreign particles are taken in by the cell through the plasma membrane. This process of ingestion of large-size solid substances by the cell is called phagocytosis, which is also known as cell eating. The term phagocytosis was coined by Russian biologist Mechnikov in 1893. Occurrence of phagocytosis is seen in most protozoas and certain cells of multicellular organisms. In multicellular organisms like mammals, phagocytosis occurs very actively in granular leukocytes and in the cells of mesoblastic origin, collectively known as the cells of macrophagic or reticuloendothelial system. The cells of macrophagic system can ingest bacteria, protozoa, cell debris, or even colloidal particles by the process of phagocytosis. In the process of phagocytosis, the external solid substances are captured by an area of the plasma membrane, which ultimately pinches off from the cell surface to form a vesicle or vacuole around the solid particle by the process named vacuolization. The membrane-bounded vesicles of vacuoles are known as phagosomes. The phagosomes, 1 to 2 micrometers in diameter, migrate to the cell interior and fuse with the pre-existing lysosomes. The food is digested by hydrolytic enzymes of the lysosomes, and the digested food is ultimately diffused to the surrounding cytoplasm. The undigested food is expelled from the plasma membrane by efflux or egestion. Endocytosis is the process of intake of materials to the plasma membrane. These materials cannot pass through the plasma membrane. It occurs by an infolding extension of the plasma membrane to form a vesicle or vacuole. In eukaryotes, two types of endocytosis can occur: pinocytosis and receptor-mediated endocytosis. the process in case of which ingestion of a liquid or fluid material in bulk takes place by the cell, that is, through the plasma membrane, is known as pinocytosis or cell drinking. In pinocytosis, the particle-free globules of fluid are surrounded and ultimately engulfed by the clasping folds of the cytoplasm. The plasma membrane forms membranous vacuoles around the fluid globules. Such membrane-bound vacuoles are called pinosomes, which are 0.1 to 0.2 micrometers in diameter. Later, the pinosomes are transported to the interior of the cell, where they fuse with the secretory granules or lysosomes. In food vacuoles, digestion of food substances takes place and the digested food is diffused to the surrounding cytoplasm. Pinocytosis can be observed ordinarily in the cells by light microscopy. In receptor-mediated endocytosis, a specific receptor on the surface of the plasma membrane recognizes an extracellular macromolecule and binds with it. The substance bound with the receptor is called the ligand. Examples of the same are viruses, small proteins like insulin, etc. The region of plasma membrane containing the receptor ligand complex undergoes endocytosis. Phagocytosis is the intake of solid material from outside to the inside of the cell, while pinocytosis is the bulk intake of fluid materials by the cell. Vesicles formed in phagocytosis are much larger in diameter than the ones in pinocytosis. In phagocytosis, membrane evaginates in the periphery to engulf solid particles, while in pinocytosis, the membrane does not show evagination. A digestive or food vacuole is formed from a phagosome, while in pinocytosis, digestion of absorbed substances may or may not occur, and accordingly, food vacuole may or may not be formed. In phagocytosis, lysosomes are essential because solid substances taken in require digestion. In pinocytosis, lysosomes play no role in utilization of absorbed materials if digestion is not involved. In phagocytosis, the undigested parts of solid particles are thrown out by exocytosis or effigy. In pinocytosis, there is no exocytosis or effigy. Thus, to conclude, we may say that bulk transport occurs across the plasma membrane of cells with the help of all three processes, that is, endocytosis or cell drinking, phagocytosis or cell eating, and exocytosis or cell vomiting.